This presentation will start by outlining how red blood cells are formed, and will then go on to talk about how the heme and the globin part of the red blood cells are recycled to maintain a healthy level of red blood cells in the blood. So starting with the process of red blood cell formation, which is called erythropoiesis. This process occurs in the bone marrow. The earliest red blood cell is called a proerythroblast, and it arises from a pluripotent stem cell which is committed to the production of red blood cells. As you can see from this diagram, this process relies on the hormone erythropoietin, which is secreted from the kidneys in adults and the liver in the fetus, and is involved in erythrocyte homeostasis. This young proerythroblast contains a nucleus. Cell differentiation and division then occurs, causing the shell to shrink, and the formation of hemoglobin in the cell's cytoplasm, which is reliant on the presence of iron, B12, and folate. This process continues until the cell ejects its nucleus and becomes a reticulocyte and leaves the bone marrow to enter the bloodstream. Around 10 to 15% of developing erythroblasts die within the bone marrow before they form their mature red blood cells. This is called ineffective erythropoiesis and is an important cause of reduced hemoglobin concentration in the blood, also known as anemia. Reticulocytes develop into mature red blood cells within 1 to 2 days after their release from bone marrow. Due to their lack of nucleus, giving them their characteristic bioconcave disc shape, red blood cells are unable to self-repair when damaged, and so after about 120 days they die. Red blood cells are destroyed by macrophages in the liver and spleen via phagocytosis. This process destroys about 2 to 3 million red blood cells every second. Hemoglobin is then broken down and split into the heme and the globin parts. The globin part is then broken down into amino acids, which is then reused for protein synthesis. The heme part is then broken down further and gets excreted or recycled. Iron is removed from the heme and gets recycled. It associates with the transferrin, which is a plasma protein, which transports iron into the bloodstream. In the liver and muscle fibers, iron then detaches from transferrin and attaches onto an iron-storing protein called ferritin. When required, iron then reattaches to transferrin, where it is carried to the red bone marrow for use in hemoglobin synthesis. The non-iron portion of heme then goes through a process to get excreted from the body. Primarily, it gets released as biliverdin and converted into bilirubin. Bilirubin is transported to the liver where it makes it water-soluble so it can pass out into the blood and it is used to form bile. In the large intestines, it then gets converted into urobilinogen. Some then gets reabsorbed back into the blood and is converted into urobilin, which is transported into the kidneys and excreted via the urine. However, most is converted into stercobilin, where it is excreted in the feces. And that concludes the red blood cell cycle.